quite nicely, is a great bridge between the basic science, the ideas, the networking, the collaborations, and then to the actual end result, which is the policy and weight. How do we get immunizations to the people that need them the most? So our third panel of the day is a great culmination of all of what we've talked about already this morning, new tools and strategies for their development, partnerships, funding, and of course, Texas research. So the three panelists are, first we'll hear from Dr. Rebecca Rico Hesse, who's a scientist with the Texas Biomedical Research Institute. And then next we will hear from Dr. Elena Bottazzi, who's Director for Product Development at the Sabin Vaccine Institute and Texas Children's Hospital Center for Vaccine Development, Baylor College of Medicine. I'd really like to see your business card, because if you can fit all of that on there, I'll be very impressed. And then the last panelist will be Dr. Bruce Lee, who's an associate professor of medicine, epidemiology, and biomedical informatics at University of Pittsburgh, and a pretty long title as well. So, Rebecca, we'll start with you first.
Thank you very much for inviting me here, Peter. I also want to thank Research America for helping us get all of us together here. Today, I'm going to be talking about the new tools that we can use or develop to study the neglected tropical disease that we've already heard about this morning. And I'm going to give you, use dengue virus as an example, because that's what I've been working on for over 20 years. So the needs or applications that we need in terms of tools, and I'm talking about laboratory-based studies for neglected tropical diseases, is that many of these diseases do not have an appropriate animal model of disease where the animal develops disease like in humans. So they're very difficult to study. You can't study mechanistics of pathogenesis in humans all of the time. So we need to spend more time developing animal models of disease that can actually translate vaccine and antiviral studies straight into human studies. Then we also have to keep in mind that numerous tropical diseases have an insect vector, as was described this morning, and that makes it more complex to study. That means that you have to be able to study the human host or the vertebrate animal. You have to study the vector, and you have to be able to work with the pathogen exclusively in these two different systems, organismal systems. That applies for bacteria and viral diseases that we've already heard about this morning. Now, the thing is that even NIH has had workshops concerning this. There are very few laboratories in the world that can handle pathogens, vertebrate hosts, and vectors. That is, to see a disease in a global, to study mechanisms in a global fashion. So the thing is that you need special containment. For example, we work with dengue virus. It's considered a biohazard level 2, but when we work with infected mosquitoes, infected Aedes aegypti, it becomes biohazard level 3. And you have to maintain all of those infected mosquitoes back in the lab. And if you're studying pathogenesis in vertebrate hosts, you have to have those mosquitoes go and bite a laboratory animal. So that becomes very complicated mechanistically. So there are very few laboratories in the world that have those systems in place. And of course, we need all of this information to be able to get more specifics about the neglected tropical diseases and how we can use the information that we are deriving from these studies to affect the models and the vaccine studies that you're going to hear about later. Dr. Murray already reviewed most of the things that I have on this slide about dengue virus, so I'm not going to repeat them, but I wanted to point out that for dengue virus, there's no other animal that develops disease. Even primates, higher primates, such as chimpanzees and gorillas, do not develop dengue fever. So that's been a problem. We don't have an animal model. So when we talk about secondary infection causing dengue hemorrhagic fever, we're not really sure. I mean, we think that's a risk factor for humans, but there's no way to study it, the mechanisms precisely, or to measure these things. The mosquito Aedes aegypti is the main vector of dengue. It lives around human houses, and it's pretty much dependent on people for its blood feeding. So this mosquito has adapted to living with us, and it has adapted to transmit dengue very efficiently. It does occur in the Gulf and eastern coast of the United States, so that's why we have had dengue outbreaks for centuries in the United States, and that's why we continue to have them in places like Texas and Florida. And we have the potential of having dengue outbreaks as far north as Washington, D.C., because the mosquitoes exist that far north. So just think of this, there's the potential there. It's a matter of which virus gets into which mosquitoes and how many humans are viremic, serving as amplifier hosts. So in terms of how dengue produces 4 different viruses, dengue viruses, produce disease in humans, it's very hard to know exactly what happens when we don't have something to test it in. We've done epidemiologic and clinical observational studies through many decades to figure out what little we know right now. And we do know that there's an increased risk when you get a second infection by another serotype. Remember, you could get up to 3, 4 different infections because there are 4 different viruses. When you get infection by the first one, you're only protected against that one. So you could still get 3 additional infections. And what we do know is that in vitro, if you add antibodies to the first virus that you get infected by to a second virus, it actually makes the virus replicate quicker in certain cells of the human immune system. So there's evidence in vitro for these things happening. There's evidence that other immune cells are involved in the production of severe dengue. That's where you have the massive bleeding and where you have mortality, which is between 5 and 10% of all the people that get dengue fever. Maybe only 5 to 10% go into dengue hemorrhagic fever. So we still have to worry mainly about what's happening with dengue fever because it's 90% of the population, which is hundreds of millions of people every year. So we didn't know that much about the virus, and we knew that the host immune system seems to produce more severe disease upon secondary infections, but we had no way of testing which viruses produce more severe disease. So over 20 years ago, we did the first phylogenetic studies of the viruses, different strains, and found that specific genotypes or specific genetic groupings of viruses, of dengue viruses, correlate with their epidemiologic potential. So right here, I have a simplified phylogenetic tree where I have viruses representing each of the other 3 serotypes down here, and then I have a group of viruses representing the West African or sylvatic cycle that Dr. Murray mentioned this morning. Those viruses don't seem to get out of West Africa, out of their sylvatic cycles. They do not seem to cause very large epidemics. And then we have another group that are from native to the Americas, And we have just a couple of viruses from the Indian subcontinent, and we have viruses from Southeast Asia. And what turns out to be the most significant correlation is that the Southeast Asian viruses that we now have across the border with us in Matamoros, this was introduced in 2005, we now have the most virulent, this is the most virulent strain of dengue serotype 2. So it is correlated with a specific genetic difference in the virus strains. So the question you had this morning about serotype 2 and serotype 3, other viruses, other serotype viruses coming in producing more hemorrhagic fever, yes, we're pretty much set up for that in Texas. This is just a protocol, a quick protocol showing what we do now. We actually transplant mice with stem cells from human umbilical cord cells, cord blood, I mean, and we can infect them with dengue virus, and they actually get a rash, shown in the bottom one. Well, one that does not contain human stem cells does not produce a rash and infection with dengue virus. Not only do they get this rash, like people do, they get low platelets, they get fever, and they get very high titers of virus in their blood. And this is just to point out the difference between the African, the Sylvatic cycle, and the ones from the Americas. I'm sorry, the West African and the Southeast Asian one. This is the most virulent strain, and this is the one that was introduced to Texas in 2005. So it produces the most disease. It also gets transmitted by mosquitoes much more efficiently, 4 to 60-fold times more efficiently than the American native virus type. So we have another advantage in the field where this virus that produces hemorrhagic fever in people can also get transmitted more efficiently, quickly, in terms of time in humans during epidemics. So these are the type of data that we can insert into the models. And lastly, this to show you what we've been doing more recently. We actually have this model. We can get these mice infected by mosquito bite. And just to show you the difference in pathogenesis and how salivary factors that Dr. Valenzuela discussed this morning are really important. If you inject the mice with a virus, they have viremia from up until about day 20. But if you have the mosquitoes bite, the infected mosquito bite the mice circulate much higher titers of virus in their blood and out until day 56. They have a harder time expelling the virus, let's say. Okay? So the salivary gland components, the saliva and the mosquitoes, have tremendous effect on pathogenesis. So that's why we have to include some of these things in our new studies. So to summarize, it's very complex. We have to study all different facets, and this has a lot of impact on vaccination strategies, which viruses to use, and also how which systems to attack first, which strains are more important epidemiologically. Thank you.
Hello, everyone. Thank you so much for the opportunity to Research America for actually allowing us to host this event here and showcase our new National School of Tropical Medicine. I think that I'm actually going to try to close the gaps of everything that we've heard this morning and earlier this afternoon. Primarily is, how do you really go from utilizing all the data that you can collect at the level of the actual pathogens, and then all the way to, like Dr. Andrews said, how do you really go into global access and actual distribution of tools? But how do you really develop the product? And by product, it can be a diagnostic tool, a vaccine, new drug. And it's, of course, very complex, and you'll see in a minute, but you really have to assess this opportunity of determining what are the real feasibility studies that you really need to understand and do to really determine if there's going to be a successful pathway of translating discoveries all the way from the laboratory and ultimately into policy and development of community interventions. So this is just to not scare you, but to just show you the dimension of the complexities of what it means to develop new drugs, vaccines, or even devices. And there's several aspects. It's a very complex slide, but there's several aspects that I want to point out. You need a lot of money, you need a lot of time, and you do also need a lot of potential candidates. If you really can see, which I also kind of circle that area, which is what we call really that valley of death, is you have to start with so many for you to maybe even be successful to get one all the way to the community, that we already in the area of the neglected tropical disease vaccine development or drug development, we may already be starting with a lot of challenges. We don't have a lot of money compared to, for instance, products that do have a lot of pharmaceutical support. And we, of course, also may not have the ability of starting with so many targets, knowing that potentially throughout this pipeline of development, things fall off because they failed for safety reasons, production reasons, clinical effectiveness reasons, and other purposes. And of course, you also need a lot of time. So this is a very long-term strategy that ultimately clearly has been shown that it's the best global health approach to prevent diseases or even treat some of the diseases by introducing new vaccines or drugs. But how we therefore in the area of especially new development, new vaccines for NTDs, how are we trying to use new strategies and models to really mitigate or try to work out of the box and try to see how we can reduce the time, save resources, and apply models where you can actually do things faster with less money, but with much more intellectual understanding of how to be successful at each of the different stages. And that's where it comes into play, this concept of working in partnerships. And these are the so-called product development partnerships. And this slide shows you that in the last 10, 15 years, certainly through a huge push from organizations such as the Bill and Melinda Gates Foundation, there has been a huge creation of different groups and organizations that really use this approach of partnerships to try to advance new tools for drugs, vaccines, therapeutics, or diagnostics. And as you can see, for instance, each of them work in different areas of diseases, each of them work in different aspects of tools. And you, for instance, can see that we are up here, the Sabin Vaccine Institute, which now is actually relocated the vaccine partnership to Texas Children's and Baylor College of Medicine. We're part of this consortium of organizations that are trying to apply this model of product development partnership. We indeed are probably the only one that actually is embedded within a medical center in an academic center. So each of them span from being very virtual and just contracting out all their services to very hands-on, where they even have their own plants and facilities. We're kind of medium. We do a lot of things in-house, but we also work a lot with partners. So what is really the philosophy? And this is where it's very important. We really combine a lot of practices that are really used in the business sector or pharmaceutical sector, but we utilize a lot of our training, scientific training and technical training that we, of course, gain out of the academic environment. We certainly collaborate with a lot of partners. We really try to evaluate this transition of the data that you obtain from the basic sciences, to develop, development strategies. Of course, apply a little bit more the understanding of how do you manage a business operation and not really a research or an academic operation. We really have to be very careful with risk management because, again, we don't have a lot of resources, and therefore every decision we take has to be really well thought out to try to avoid making many mistakes. And we learn a lot from, of course, others' mistakes, so we try not to make them. And create a lot of flexibility in our decision making, really establishing those criteria that would determine if it's a go or a no-go, and really be more strict in what to move forward during the pipeline. So to make it even more complicated, we even ask us many, many questions. And usually what you end up really doing is asking questions from all the way from the end, and then we kind of go backwards and see how do you go by knowing what the global impact of a certain disease is. Does it really address a public health need? What is the significant global burden? How do you really even go by, determining how will be this global access plan? Even already thinking, how would a country introduce a hookworm vaccine? If we don't even understand what are the different potential models and start working with the governments to start, preparing for that, we wouldn't even know the type of vaccine that we actually have to produce, because it has to be, produced in a way that would fit eventually how the introduction of these vaccines should be done. Of course, then we use pretty much the pillar, which is the R&D aspects, where we really have to understand, like you heard, throughout the morning, where is the burden, the epidemiology, what are the models already in the laboratory we could use? Is there any, ability of doing any studies in human populations? Do we have even mechanisms that we can predict protection? A lot of immunology. So you utilize all the data that comes out, mostly from the academic sector, to then really determine if we could set up very specific preclinical studies where you can actually then determine efficacy. And then it goes into the very burdensome portion of the development pathway, which is all the product development and the clinical development. And this is just a snapshot of really showing you just briefly what it entails to actually do product development. So from that discovery, you really have to go through a series of steps where you have to do feasibility for expression and production and scaling, but there's a huge gamut of quality control without not even being close to entering into the clinical phases, which is really the next step that you wish to try to accelerate. So this is exactly what we do here in the labs. We actually bring these discoveries, we try to design mechanisms to make them in the lab, scale them in the lab, and then eventually transfer them to institutions that actually make them for human use. And that's just the product component. But what happens to, for us to be able to influence and accelerate the clinical development, which is ultimately what you really want. And this is what we use. We use a rapid into phase 1 model, which utilizes the ability of very rapidly testing these different products in the clinic, because generally that gap that I showed you, which is that valley of death, is that most generally products fail when they enter safety testing. So we try to mitigate, the ability of recognizing which candidates actually will continue through the vaccine pipeline development. So in summary, this is really just a very brief snapshot of how complicated it is, but I want to highlight that, for these PDPs to be really successful, it takes some time to build. Then you really have to leverage the expertise, and eventually, you're able to sustain them, which is the importance. But as you can see, the key strategies are partnerships, certainly doing a lot of training and capacity building, working directly within the countries that you really want to eventually see these products being introduced, working with organizations such as governments, PAHO, WHO, because they really help you in this global access component. So a lot of advocacy, a lot of proof of performance milestones, and ultimately that really is what eventually you can use all that data to obtain the sources of funds to really make the product really advance all the way to the delivery and distribution. So I thank you, and we can open to discussion after the panel. Thank you.
All right, thank you. So I wanted to thank the organizers of the conference, Research America, as well as Baylor College of Medicine and National School of Tropical Medicine, for inviting me and this opportunity to participate with this distinguished panel of speakers and guests. So I'm going to be talking about economic modeling and simulation, basically to evaluate new technologies, specifically technologies that will serve in the fight against neglected tropical diseases. As was echoed this morning, we have an opportunity to have an open discussion with policymakers and decision makers about next steps in terms of the control of these neglected tropical diseases. But it's helpful to have tools and an armamentarium to initiate these discussions and continue these discussions. Also, as Maria Elena alluded to, there is this potential gap between technology development and market needs that needs to be closed. And I propose that modeling and simulation is one methodology that has been used in the past and has a tremendous potential to close that gap. So just to start, this is my research group, which focuses on developing computational models and simulations that address both economic and operational concerns in the control of infectious diseases. So the neglected tropical diseases is one of our focus areas, with Chris Bacon leading our efforts with that. So economic modeling can serve as a bridge between all aspects of technology development as well as technology translation. So economic modeling can help design appropriate preclinical and postclinical studies, as well as epidemiological studies, and take the results of those and translate to policy and practice. So there are numerous stakeholders that can benefit from economic modeling, first of which are product developers. So in establishing target product profiles, as many of you are familiar, these are a menu of characteristics that a technology, for instance, a vaccine, should have, and it's almost like a wish list, saying that it should have a certain efficacy, a certain duration of action, a certain target population, even potentially price points as well. Now, TPPs have been used widely in discussions between product developers and regulatory bodies such as the Food and Drug Administration, but many times in the past, these TPPs have not benefited from modeling, because many times the wish list is determined in conversation between the regulatory body and the scientists or developers, but its potential effect on the market is maybe not clearly elucidated. One of the things with vaccines, as all of you know, vaccines have been one of the most successful public health technologies in the history of public health. But one of the challenges has been that vaccines effectively are supposed to be pull technologies. By pull technologies, I mean you have a market need, and vaccines are developed to fill that need. So you have a certain disease which is spreading, and you need a vaccine to curb its spread. However, many times product development with vaccines occurs as a push technology. Someone finds an antigen and starts developing the vaccine as a result. Sometimes the vaccine will reach the market and may not perfectly fit the market needs. There are many examples in public health in which a vaccine has reached the market and maybe faced initial struggles because there are certain characteristics of the vaccine that didn't quite match the market. For instance, with the example of flu mist, where the vaccine was priced a little too high, or an example of the rotavirus vaccines, where some of the initial packaging was a little too large to fit into the supply chain. So different aspects like that. So if we can use modeling to help define these TPPs in advance, when you can actually readily fix the characteristics, this might help the market anticipate the arrival of the vaccine. Product manufacturers, similarly, have many manufacturing, pricing, marketing, and distribution questions. So if you develop models early on, it can help manufacturers fit these needs. Third-party payers are very interested in coverage and reimbursement issues. So you can have economic models help identify the potential economic value of a vaccine and where reimbursement points should be set. Similarly with funders. So major funders are always asking, well, should I fund the development of this vaccine? What's the potential return on investment? What should be the portfolio allocations? For instance, if I'm sitting in a foundation and I want to allocate a certain percentage of my portfolio to vaccines versus drug treatment versus other preventive measures, and policymakers as well. So they have many budgeting questions, and they want to understand how to implement vaccine programs. The one category that I didn't neglected to add here was also businesses. So many times, various businesses can benefit or can be impacted by the spread of infectious diseases, and they may not realize the benefit of a vaccine. Examples of that include the flu vaccine. We did a study in which we showed that employers actually benefit in terms of their bottom line, their revenue, their cost, and their profit if they ensure that their employees are vaccinated with the flu vaccine each year. There have been studies which have shown that administering antiretroviral therapies to laborers in various locations, such as South Africa, can actually significantly benefit companies who employ these individuals because their productivity rises. So many times you can actually identify benefits for different stakeholders, benefits that they may not have realized if they didn't see an economic model as such. So let me give you a few examples. For instance, the economic value of a dengue vaccine. So we published this in the American Journal of Tropical Medicine and Hygiene. So essentially what we did was we constructed a model in which represented all the different possible states that occur if someone became infected with dengue. So the model essentially sent thousands of individuals through this simulation, and some of them became infected with dengue. Of course, their chance of infection depended on whether they're vaccinated or not, and if they're infected with the virus, then they have probabilities of developing different sequelae, and each of those sequelae could generate costs. So here are some of the summary results, which show, for instance, this top graph shows a dengue risk of 5%. This is a dengue risk of 9%. So these are two different scenarios. And along the x-axis is the cost of the vaccine. Along the y-axis is the incremental cost-effectiveness ratio, which is essentially the ratio of the cost for the situation in which you have the vaccine minus the cost where there's no vaccine over the health effects, which we measure in DALYs, disability-adjusted life years, of the vaccine scenario minus the no vaccine scenario. So the WHO has defined something as being cost-effective if its ICER falls below 3 times the GDP per capita for that country. And it's highly cost-effective if it falls below 1 times the GDP per capita for that country. In this situation, we looked at Thailand, so this is the GDP per capita for Thailand right here. And we found that if the vaccine cost is under $100, in most cases, it's highly cost-effective, and in many cases, it actually becomes cost savings. So it can actually save costs by vaccination. These different curves are different vaccine efficacies. And again, this is the dengue. If you raise the dengue risk, all of these curves start shifting downwards. So the main take-home point with this is that you can have a wide range of vaccine costs, even up to, in some cases, $100, $200, or if the risk increases, even greater. And the vaccine can still be highly cost-effective, and in many cases, cost savings. Also, the efficacy, these hold within a wide range of efficacies. So another take-home point is that you don't have to have a necessarily perfect vaccine to have it be economically valuable or even cost savings. Of course, everyone would want to have a vaccine that's 80, 90% efficacious, but even if that target is not reached, there's still potential substantial value. We did the same thing with a Chagas vaccine. This was published in PLOS NTD. And this chart, this table shows vaccine cost along here, different vaccine efficacies, and different TGRC infection risks. The main take-home point with this is there's a large number of scenarios in which vaccination is highly cost-effective and, in fact, many times cost savings. This shows similar scenarios, but we're actually looking at just the cost. So what's the incremental cost of vaccinating an individual? So every time it's negative, you're actually saving costs. So in this case, for instance, if you vaccinate an individual, you're saving $2 with a very low risk, relatively low risk of T. cruzi infection compared to the other scenarios, and a low vaccine cost, but a low vaccine efficacy at 25%. Sometimes showing these numbers can be effective to certain stakeholders because showing the cost effectiveness ratios may be useful for larger policy, but if you want to demonstrate to a stakeholder what's the potential budgetary impact of implementing the vaccine, to show them that they can actually save costs is quite substantial. This is actually rare compared to, in the whole span of medical interventions. Most medical interventions require some type of investment or some type of outlay to achieve health benefits. When something saves costs as well as achieves health benefits, it's considered economically dominant. It's a no-brainer. In other words, you really aren't expending anything to gain health benefits. So in the interest of time, I'm going to skip through many of these slides. We've done the same thing with Leishmania vaccine as well. One thing that we're consistently finding with various neglected tropical diseases is there's many situations in which vaccination is highly cost-effective or cost savings. And that's because with these set of diseases, their impact, their economic impact is so great on populations that you're actually averting that with vaccination. And even if you're averting only a small percentage of the cases, you're still potentially generating cost savings. The other take-home point is that unlike diseases which may have very high mortality, many of these diseases don't necessarily have high mortality, but they have a high economic burn. In other words, they cause loss in productivity, they cause chronic problems which may not be easily measurable. So it's easy to garner headlines when you say this virus is going to cause millions of deaths worldwide. That seems to grab policymakers, especially if those deaths are in Reston, Virginia, for instance. But on the other hand, those diseases, many times they have a very quick burn, and you have deaths, and otherwise you don't cause as much chronic problems. But where you have like Chagas disease, hookworm is another example, you basically have a slow burn of resources that can fall beneath the radar for the media or for policymakers, but in the long run actually can cause a much more substantial burden. So it's important to many times to put that in dollars and cents, because that's the language and that's the currency, literally the currency that many policymakers and stakeholders speak in, whether you're talking about manufacturers, developers, funders, or policymakers. So in conclusion, economic modeling can assist a wide variety of decision-making, from product developers all the way to policymakers. New technologies can bring hidden economic benefits, perhaps even paying for themselves. So if you can demonstrate the return on investment is very positive within a short period of time. So we recently completed a study which is going to be published in Human Vaccines, which shows that you actually recoup the investment into a therapeutic Chagas vaccine within the first year of rollout in many scenarios. So that potentially can be strong motivation for additional developers and funders to enter this area. And finally, technologies to prevent even quote-unquote low-risk neglected tropical disease can be valuable. I showed in some of our previous models how we varied infection risk. So if you take that risk, and certainly in certain high-risk countries or locations, the vaccine can be very, very valuable. But you can find that even in quote-unquote lower-risk areas, for instance, we're seeing cases of many of these diseases in Texas, and it's still considered low risk in certain areas, a vaccine or other types of treatments could be quite valuable. So be happy to entertain any questions as a panel, and thank you for your attention.
Thank you. If the other panelists will come up to the front. I have just a couple of minutes for any additional questions or comments. I would have a question the product developed partnership is relatively new conversation in these last years. You're hearing it more in policy circles in Washington. How are you, are you feeling that this is getting traction with the people that should be paying attention to this?
I think it is getting traction, and I think it's more importantly not only getting traction here, but also within the developing countries that we know that eventually will really have to deploy these types of vaccines. So the typical example is, in fact, we've been working with the Brazilians practically for the last 12 years. And we've been working with them to educate them on the importance that this is a partnership, that they're actually owning the project the same way, that it's not just us developing on our own. And recently we've really had some great milestone where they now are investing themselves even in the development of, for instance, the hookworm vaccine in this case, or schistosomiasis vaccine. So I think it's really showing that the value of working within these partnerships, which is just bridging not only the technical but also some of the policymakers throughout early in development, knowing that eventually they will apply it, within their own policies of, vaccine distributions.
And then it sounds like that if your Brazilian partners are picking up on how this can benefit them, we can also use that as kind of some learning modules for reaching out to other partners in other countries.
And the typical example is then that now we're just starting to work with the Mexicans because they've seen now the track record of how important it is to not only wait until the vaccine is developed and then figure out how they could potentially introduce it, but actually now they're interested to really start working with us from very early on in development, thinking that eventually they would also be able to establish these mechanisms within, let's say, Mexico for the Chagas vaccine or Leishmaniasis vaccine.
Okay.
Actually, I want two questions.
Fine.
So one question for the panel, and then one question for John Andrus. Question for the panel is, are products for the diseases that we're talking about today, for neglected diseases in Texas or in poor parts of the U.S., are they going to fall through the cracks for funding? Because they're not a priority for the Gates Foundation. The remit of the Gates Foundation is Sub-Saharan Africa and Asia and maybe a little bit Latin America. The remit for the NIH has not typically been these neglected tropical diseases. They've done a good job funding basic research, but not really products, nor for public health. So we're not here, we're not there. We're in somewhere in the middle.
Peter knows my answer.
Well, my observation is, I certainly agree with your observations that we look at some of the major funding bodies and they don't immediately recognize the burden and potential benefit of these treatments or preventive measures, and that many times it's a post hoc situation where the problem reaches a certain level and it becomes prominent, and then suddenly there's a rush of attention and funding. So the hope is to demonstrate the hidden, in this case economic or burden, economic impact that these diseases may have, and I think part of it is an educational campaign or a marketing campaign to understand that, but there's no easy answer. But there's been many situations in which this education or speaking the language of decision makers, I think, is key. And eventually there's a realization of the potential burden.
I think we're at a historic opportunity to have that message heard in Washington, for sure.
And then, thank you. And then my question for John is, John, you've by now have probably been to every Latin American country and seen poverty and disease in every Latin American country. what would you do now? What if your portfolio now included South Texas and you were visiting South Texas as a poor Latin American country? What would you do?
Can you hear me? Yeah. As a worker or an immigrant?
As a public health expert.
Yeah.
As he chose got his hands.
Yeah. Yeah, I was, that's a great question. What comes to mind happened yesterday, actually. I was honored to be able to introduce Professor Sir Michael Marmot, who's done a lot of work with the social determinants of health at the Pan American Health Organization. And he, at the end of his talk, outlined the way forward. I mean, the social determinants of health are so cross-cutting, and they've been mentioned throughout, they percolated throughout virtually every talk here today. But he mentioned values. So he mentioned 3 or 4 things that come to mind. Values, and values that have to do with equity, access. Those are values that, coming from, in this scenario that you asked, perhaps are more prevalent in Latin America in some cases, I believe in a lot of cases, in comparison to what we have in the United States. Because there are universal access to vaccines in most countries of Latin America. They're provided for free because it is recognized as a global good. And there's, in a lot of countries, excellent examples of a safety net for those that live in poverty. But having said that, it's the region of the highest disparities as well. Political commitment was another thing to work on that I think many here in this room are trying to champion. The third thing that I recall was the community involvement. So that's where my angle would be on this, in this situation. Begin to work with the communities to recognize this as a problem. I think he was saying it more in terms of recognition, but also implementation of the action and the mobilization of the community voice that would help drive other forces, like the issue of political commitment. At the community level, also evidence. Evidence in general was something he highlighted that I think applies in this situation. So whatever I could do at the community level to ensure there was evidence. And then over time, monitoring. I think those were the key messages that I think are guiding principles of all our work. But at the community level, if that's where I am, that's what I need to do. And you know, what is it? What's the old saying? Local action with a global vision. Or, but I'll stop there. I would have loved the opportunity to take the local experience and then be able to go up the chain of authority to take the evidence up the chain.
All right, thanks. Let's just go to Joe, and then to you.
I want to reiterate 2 things that John said. One is the issue of equity, but equally importantly, the community involvement. First of all, the marketplace is not interested in equity. It's interested in making money. And we've learned from, if anything, about vaccines that, at least I have, that the marketplace has failed when it comes to having equity for vaccines, which is why we have the Gates Foundation and others stepping in to intervene. And I think part of that is because in this country, even if the marketplace is not providing total equity to everybody, we have enough voices that we can push that. In other parts of the world, those voices are not there. And that's why the community issue is so important. In South Texas, we see this because there are huge disparities, health disparities, enormous ones in South Texas. And there has not been a voice. Only in recent times when the NIH created the National Institute of Minority Health Disparities, and there's been more discussion, presumably because people understood that our social fabric was going to be literally destroyed if we don't do something about this, that things have started to move a little bit. But there are still these enormous disparities. So I have a question for Dr. Lee, and that is, should vaccines disappear out of from the market and become some kind of an international program that basically ignores the market and says we have to have equity and justice, and therefore we have to, even if it's the poorest. I used the example in my talk about the meningitis belt in North Africa. It took 25 years, 30 years, despite the fact there was an available vaccine. And I sat in meetings where people said, we're not going to make this vaccine because nobody can pay for it. Nobody being, we have hundreds of millions of people across North Africa, but presumably they're nobody because they can't pay for it. And now on the other hand, people argue, well, if we don't create a marketplace everywhere, then we'll continue to give away vaccines, and that'll just become part of what we do, and it's costly and inefficient and so on. So I'd really like to hear your thoughts about this as an economist. How do we, I mean, is the way we're doing it, addressing it with the Gates Foundation and others, basically footing the bill? Is that the way we're going to continue to do this, or is there a place for a true market economy in global vaccine business?
Well, excellent points and question. So this calls to mind a term that I believe was coined by Bill Gates when he called it creative capitalism. So one of the things that he's emphasized is that there's been a disconnect in many cases, especially in global health, between the need of the marketplace and manufacturers to fill that need. So he argues that in the perfect world, you have a need. For instance, if people need to eat some vegetables, you have a vegetable seller, and that vegetable seller will make money because he's filling a need. If someone is selling popsicles and there's no need for popsicles, then that person will then shift over to vegetables. But he's argued that because there's been a lot of things thrown in between the buyer and the seller, especially in the global vaccine market, that's been twisted around. So what he has proposed, and some others, has been trying to narrow that gap, bring that together, and remove those forces that are intervening between the buyers and the sellers, or come up with innovations, economic innovations, ways to, well, the first thing, of course, is to show that there are potential gains that may not be obvious. So I think historically people have said, okay, well, vaccines, low margin. Low margin, you're giving vaccines, it's not a recurrent drug or medication. So it's a low margin, you give it to the population once, and then you're done, and you're not going to make much money, as opposed to, say, a chronic drug, chronic medication. The other problem, some manufacturers have argued, is you can't expand the market. So you can't convince someone, well, instead of one vaccine, why don't you take 5, or take 10 doses? Whereas certain other medications, you can expand the market. But I think the first step is to show that there are opportunities there. I mean, there are differences, there are advantages that vaccines have over all those medications as well. I mean, you have an installed base, you have the support of countries and major organizations, and I can go through a laundry list, but in the interest of time, I won't. But one thing is to emphasize those advantages and potentially construct models to show what those advantages are. Second is potentially developing new mechanisms to bring the buyers closer to the quote-unquote sellers. I think if you remove it completely from the marketplace, you do lose some of those advantages. Of course, that's a continuing debate. But I tend to shade towards the side of the arguments that Bill Gates is pushing, that you can use creative capitalism. You just have to think of new mechanisms.
Thank you. We'll get the one last comment.
It's actually related. Just thinking that we live in a global economy now, that we're expanding or we're wanting to expand, and at the same time, finally, we're realizing about the importance of the loss of productivity. So I see that many corporations are waking up to this, and they're making coalitions to try to respond to things like CAMA, an example for malaria. I was just thinking, is there any possibility not just to bring the communities into the picture, but if there's not, the way to convince them, create the need for corporations through maybe a kind of taxation or something, that if you do want to work in Nigeria, you do need to respond to this public health. And after you do this, the best bid is the one that's going to open the oil exploration or something like that. Is there a way to bring finally that? Because we're growing into a global economy. I mean, this is where we're going. We have disasters. We're not getting any better in our world. So we will need to expand to those countries, or we will have them coming to us. So we will have to deal with these diseases in the future. So at this point, is there a way to act in a more aggressive way? Taxation, something like that. That's my idea.
Yeah, this in some ways echoes a conversation that John and I had over lunch, where all these things are linked. So businesses are linked to the communities around them, not just the communities around them. We're having more of a global marketplace. So what's going on in certain parts of the world is going to affect businesses throughout the world. And we are starting to see examples where we can show, if you control this type of disease, and I gave the example of HIV control in South Africa, where that was actually a hidden burden on many of the businesses in South Africa. When they realized that that was true, they were willing to pitch in to help with antiretroviral drugs. So there may be other opportunities, which goes in line with the creative capitalism, where you can actually demonstrate that this policy, like you mentioned an example, could actually help bring in other people to help fund the control of these diseases. I think the key is the equity argument works to some degree, but the challenge is there's a utilitarian. You have to come up with a utilitarian argument for many decision makers. You have to convince them. Of course, many decision makers will say, "That's great. I want equity, but I have to consider my bottom line." But if you have both arguments, if you say, okay, your bottom line is actually going to be improved if you implement this or if you chip in, then I think we can potentially find win-win situations. And I think that's one of the next steps that may be helpful to pursue.
I think that's part of the creative capitalism that some of the science community can think about as well.